This conference will now be recorded.
Hello students, I'm Dr. Divya from TeenEinstein. This is the first class of 10th chapter. So we are moving to 10th chapter. Microbes in human welfare. Microbes, have you heard about microbes? What is microbes actually? Microbes means they are single organism. Okay, they are single celled organism and we cannot see them with naked eyes. Okay, so what are microbes? Microbes are single celled organism and they have very minute structure or they are invisible to the naked eyes. Microbes means, so microbes, what do you mean by microbes? They are single, majority of, not all the microbes are single cell. Single cell, generally we say single cell. They are invisible. Invisible. We cannot see them. See. Okay. Minute structure. So they can be seen under microscope. Under microscope, we can watch all these organisms with higher magnification. So with our naked eye, it's impossible to see these organisms, and they are also called as microbes are also called as microorganisms. Micro means very minute structure. Microorganisms. Okay, microbes are also called as microorganisms, and there are different types. You know, there are different types of microorganisms. What are they? Or microbes. There are different types. In 11th standard, hope you have learned about different kingdoms of organisms. There are different types of organisms which belongs to different kingdoms, like plant kingdom, animal kingdom, then kingdom, Monera, Protista, everything you have learned. So there are different forms and different sizes of these organisms. For example, for microorganisms, which and all we can say fungi, virus, bacteria, protista. Then all these are belonging to microorganism. Example, bacteria, fungi, then protista, virus, all these belongs to microorganism. See, what are microorganisms? They are simple organism or simple structure which is having minute structure and we cannot see them with naked eyes. Okay. So these microbes, these kind of microbes, they are useful. Some organisms are useful and some causes harmful effects also. Some organisms are very useful and some are very harmful also. So they may cause infection. Harmful organism, harmful microorganisms, they may cause infections and some kind of diseases and all. That's why we are calling as harmful organism. So and some other microorganisms, they spoil food items or food materials, they may spoil. But some are helping. Right? Some helps in the maintenance of the environment also. And you know, some bacteria are helpful. So in this chapter, what and all we are going to learn, we are learning about the role of microbes in household products. In household products, actually in this chapter, we are going about the positive sides of this microbes. They're useful. We are learning about the useful microorganism or how we are making use of this microbes and their activities, how we are making use of their work or their body structure, or how we are making use of these microbes in daily life. That's what we are going to learn in this chapter. There are some harmful, we have discussed about the diseases which is caused by these organisms, like bacterial infection, fungal infection, everything we have learned. So this is the positive side or the useful side of this microorganism. So we are learning about the microbes in household product, then industrial product, in sewage treatment, then biogas production as biocontrol agents, how they are working, then biofertilizer, in that how these microorganisms are working. All these things we are going to learn in this chapter. Okay. So you know that these microbes, they are the major part of this biological system, because you have learned in class 11 the diversity, how these organisms are diverse, and diverse forms of these organisms you have learned about. Like I have told, different kingdoms you have learned, right? In that, kingdom Plantae is coming, kingdom Animalia is coming, and that and all, it is having very big structure. We can see with our naked eyes, right? But what about in the case of this microorganism? It's not visible. Right? They are present in soil, they are present in water, air, then inside our body, in our intestine, some organisms are present. And in some cases, we can make out one special feature about microorganisms where other life forms, they can't exist. There also these microorganisms can exist. In some other organisms like microorganisms, we can see other life forms cannot exist in some particular places, but there also these will be present. See, inside our body, some are present. Like in thermal vessel, you can see some kind of microorganism where the temperature is very high, or where it is above 100 degree. Okay, and it is deep in the soil. There also this like gives us thermal vents and all. Some kind of microorganisms are present which can tolerate that high temperature. That's why I said in some places where the life forms cannot exist. Can you imagine about a human being at 100 degrees? No, it's impossible. But there are some organisms, microorganisms, which can persist under this high temperature condition, because that is having that temperature tolerance. Okay, so these organisms, they are present at extreme conditions also, and some other organisms, they'll be present at in extreme cold condition also, under snow and all. So you can see different types of organisms. I said protozoa, bacteria, fungi. Then some may cause infection, some may cause, or some may be helpful to human beings, or some may have positive sides and others have this infection. Others may cause this infection and diseases also. Okay. Here you can see the organisms like which I have marked here. They are bacteria. These are bacteria. So we have learned about different types of bacteria with different shapes. See, these are, here you can see this. These are rod-shaped bacteria. Then this is spherical shape, rod-shaped. This is spherical shape, and sorry, here you can see the down one. That is a magnified one. Down you can see the rod-shaped bacteria with flagella and all. It is magnified. Here you can see the flagella. This is under magnification. Under that high magnification, you can see the complete structure here. This is rod-shaped bacterium. It is having flagella. Okay, here the flagella is marked. These are the flagellas. Okay. So there are different shades. I'm saying about the different shades of bacteria itself. You can see different shapes. We have learned about this. Then next is viruses. The first one is the bacteriophage. This is a bacteriophage. Okay. Here, the structure of a bacteriophage. Here you can see the head, then a collar, the neck shape, the collar one, then tail you can see here, this portion that is a tail, then a plate structure you can see here, then prongs, these are prongs. So this is structure of a bacteriophage. Then here you can see adenovirus. This is causing respiratory infections. Adenovirus. Okay, this is the adenovirus which is causing the respiratory infections. Then here, this is TMV, tobacco mosaic virus, which is rod shaped. And this is under high magnification. You can see the compact rod shaped viruses, TMV, tobacco mosaic virus. So, you can see here the colonies. What is this? The colonies of microorganisms. This bacteria, fungi, and all you can see as colonies, and they can grow in nutritive media also. So this nutritive media, which in that also these bacteria and fungi, they can form colonies. See, these are the colonies that group. Okay, here you can see the fungal colony. So when we will use this for studies, we are using this kind of in Petri plate. These are Petri dishes. In Petri dishes, you will pour nutrient media, and in that nutrient media, these colonies will grow. Okay, there are different methods to make these colonies. Okay, and in food items also you can see when it is getting spoiled, you can see fungal colonies on that, green or white patches like powdery appearance. You can see the fungal colonies, right? And we said microbes, they may cause different diseases, also infectious diseases, and in some other cases they are very useful to human beings, and they may they are used in industries, then agriculture purpose they have used, then at home, you know, batter, we are using dosa batter, idli batter, everything for that and all. Okay, for that and all, microbes are becoming very useful. Okay, so they are very useful in some cases, and at the same time they may, some other microorganisms, they may cause infectious diseases also, but all belongs to the same group, microbes. Okay, so now let's see from this chapter you may get neat questions also. So for that neat questions, after completing all these topics, I will explain you the extra point which is for neat exam. So in chapter we have learned about different diseases which is caused by these microbes, right? And they can cause, this microorganism can cause diseases to plants and animals. But you cannot think that these are harmful always. So some organisms, just now I said some microorganisms, they are helpful in different ways. They are helping in human beings. So the major contributions or the major useful things about this microbes that we are going to discuss now, or where and all, in which field and all we are making or we are utilizing these microbes. So first we are going for microbes in household products. Okay. So first we are discussing about in household products how it is becoming useful. So every day we are making use of this microbes in the household products, right? So which is a common example? Common example is production of curd from milk. From curd, we have production of curd from milk. Okay. Or Don't get confused. We'll write it as from milk. What is forming? Curd. Okay. So from milk, curd will get produced. So microorganism like lactobacillus, which is helping for this, the microorganism which is helping us, lactobacillus, which is the microorganism, and commonly they are called as lactic acid bacteria. So this belongs to bacteria. Okay, lactobacillus, it's a bacteria, and the group of bacteria which is helping to form curd from milk, they are called as lactic acid bacteria. Lactic acid bacteria. So in short form, we can call as lactic acid bacteria. These can grow in milk and they can convert the milk into curd. So lactic acid bacteria, this group of bacteria can grow in milk and they can convert milk into curd. Okay. So this, from the name of this bacterium itself, it's easy to find out. For exam, it's easy. Lactose, it is related to milk. So lactobacillus, which can grow, this is a group of bacteria which is generally known as lactic acid bacteria, and this can grow in milk and that can convert the milk into curd. Okay. How it will work? This lactic acid bacteria that will produce some kind of acids. So during their growth time, this will produce some acids. Okay. These bacteria produce some acids and that will coagulate the that can coagulate the milk and that can partially digest the milk protein. Digest milk protein. Okay, that what it will do, it will partially digest the milk protein and that can produce some kind of acids which can coagulate milk into curd. And how we will make this? How we will make this curd? At home, usually how we will make? We will add small quantity or a less quantity of curd. Right? Less quantity of curd we will add to fresh milk. This will be added to fresh milk. So to fresh milk, what you will add? A less quantity of curd that we will add to fresh milk and that will get converted into curd. How it will that less quantity that you will call as inoculum. Inoculum. This is the inoculum or the small quantity which you are adding. Because why you're calling it as inoculum? This contains microorganism that curd, the small quantity of curd which you're adding to the fresh milk, that is containing the bacterium, lactic acid bacterium. That group of bacterium or lactobacillus is present in that inoculum. So that will be added to the fresh milk. Then the fresh milk is getting the bacterium and that bacterium may produce some kind of acids which can coagulate milk and that can digest milk protein. So it will get converted into curd. Okay. This so small amount of curd is added to the fresh milk as inoculum. Don't forget this word inoculum, and that fresh milk, whenever this inoculum is added, which is having the bacteria, it will start coagulating. That will produce some kind of acid and that will coagulate the milk and digest the milk protein. Then it forms the curd. So this inoculum will be having so many lakhs of this bacteria. Okay. It is very easy, the division of that bacteria. So it will be having so many lactic acid bacteria. Okay. And when the temperature is suitable, when the temperature or the conditions are suitable, then this bacteria will multiply and that will convert the milk into curd. Okay. So when this milk is getting converted into curd, when it is converted into curd, the nutritional quality is also increasing. Why? When it is in curd form, it will be having vitamin B12. Okay, vitamin B12. So when the milk is getting converted into curd, the nutritional quality is also increasing. Okay. Nutritional value is high in curd. Okay, nutritional value is high because it is rich in vitamin B12. Curd is having vitamin B12 content also. So in the stomach, in our stomach, this bacteria, lactobacillus bacteria, it is playing a very good role, or it is having a beneficial role in human body, and that can prevent other microorganisms also, that can control or check or prevent other microorganism. Okay, this lactobacillus can check other microorganisms. Okay, so what and all we have learned? When milk is getting converted to curd, with the help of which bacteria? Lactobacillus. Lactobacillus is a bacteria. That is a bacteria. So that belongs to the group of bacteria. Lactobacillus. You have learned about different types of bacteria. With the help of shape, you can make out bacillus, coccus, right? Like the different shapes we have learned. Bacillus means the rod-shaped bacteria. Okay, so the lactobacillus, which is belonging to curd. So when milk is getting converted, which bacteria will work? When it is becoming curd, lactobacillus will work. This lactobacillus, it is a bacteria, and when it is growing in the milk, a little bit curd that is added as an inoculum, which is having thousands and millions of this lactobacillus bacteria. That is a lactic acid bacteria that belongs to lactic acid bacteria group. So when that little small curd, when it is added to fresh milk, that will be having a kind of acid which can coagulate the milk and digest the milk protein. That will partially digest the milk protein. Okay. So when milk is becoming curd, the nutritional value of curd is high compared to milk. Okay. So the curd is having vitamin B12 content also. The nutritional value is high. Okay. And the small quantity of curd which is added to fresh milk, that is called as inoculum. Not only this curd is called as inoculum, whatever that bacterial content, that part which we are adding to make some useful product, that is called as inoculum. Okay. So lactobacillus is having very good beneficial or a very beneficial role in human body. Okay. Then next is what? Next is about dough. Dough, this dosa batter, idli batter, everything you have seen. So that is also fermented by fermented by bacteria, that is another bacteria. So you can see that if grinding and keeping the batter morning, it will be somewhat puffed appearance will be there, right? So that puffed appearance of this dough is it's due to the production of what? Production of carbon dioxide. Carbon dioxide gas is forming inside that. That's why it is forming. It's forming that puffed up appearance. So you can see that the preparation of rice and black gram. We are using the urud dal or black gram, that is used for the idli batter, right? So microbes, which and all microbes are helping? Streptococcus species. Okay, Streptococcus species, it is helping Streptococcus. See, this is another shape, right? Here we have learned about bacillus. This is coccus. Okay, so Streptococcus, these are all helping for fermentation. Okay, the dough, dosa, idli batter and all, you have seen the puffed appearance is due to the presence of carbon dioxide formation in that when we are grinding the urd dal or black gram plus this what rice. We are grinding it and keeping. The next day it will get fermented and the puffed appearance will be there. That puffed appearance is due to the formation of carbon dioxide gas, and which bacteria is helping? Streptococcus species of bacteria. And one more, leuconostoc. Leuconostoc and Streptococcus both are helping in this. Okay. Then another one is in bread making. So bread, in bread making, what you can see is okay. Next is in bread making. In bread making, which one is used? Next is bread making. This is of microorganism. In bread making, what and all you can see bread making, that is a common thing. You can see so many YouTube videos. We are making this type of cake, that type of cake, vanilla cake, pineapple cake, like that. So bread and how to make bread, bun, all videos are available, right? So how it is formed or how they are making this bread or cake. So in everything, in such things you can see fermentation. It is done using baker's yeast. What is used there? Baker's yeast. Baker's yeast. So while adding or while making this bread or bun, we can see that in that dough, what we are adding? Yeast. Yeast we are adding and keeping it. Right? So baker's yeast, which is that microorganism Saccharomyces cerevisiae. Saccharomyces cerevisiae. Okay, Saccharomyces cerevisiae. This is the organism. This is called as baker's yeast. Okay. So when bread is prepared from dough, it is fermented using baker's yeast, which is called as Saccharomyces cerevisiae, and there will be a puffed appearance of dough. It is due to the production of carbon dioxide. In this also, carbon dioxide is produced during fermentation period, and this carbon dioxide gas during that baking time. Then after making this bread dough, after that we are keeping in the oven or something, cooker oven and all we are keeping, right? So that time this carbon dioxide gas with ethyl alcohol that will be evaporating during that baking period. So that makes the porous and soft form of that bread or cake. So you can see this is a bread piece. Means you can see a porous appearance, right? So through this pores, it is the carbon dioxide gas is going out. Carbon dioxide along with ethyl alcohol, because there it is getting fermented with the help of the bacteria Saccharomyces cerevisiae or baker's yeast. We are adding to the dough. So their fermentation process is going on. So after fermentation, which gas will come out? Carbon dioxide. So carbon dioxide along with the, which one? Along with ethyl alcohol. Carbon dioxide along with ethyl alcohol that will evaporate during the baking period, and that makes the soft and porous appearance of that bread. Okay. Then another one is toddy. What is that? One second. Next is toddy. So what is toddy? It's a drink. It's a traditional drink in South India, and it is made by in South India. You can see toddy shop like that, boats. Okay, it's a drink which is made in South India, and how it is formed? It's made by fermenting sap from palm. It's a traditional drink which is made from palm. Okay, palm. Which palm? The scientific name. Okay, Caryota urens. This is a scientific name. So these things and all you should note down. See, for board exam, if you are writing traditional, it is a traditional drink which is seen in South India, and this is fermenting by the sap of palms. The sap of that palm, it is getting fermented with the help of which one? With the help of naturally occurring here. Which process is going on? Fermentation. Okay, this sap is collected into pods. Some pods it will be getting collected, and the next day it will be collected. So the next day it will be getting fermented with the help of which one? Naturally occurring. Which microorganism is fermenting it? Naturally occurring yeast. Again here yeast, but this is naturally occurring yeast. It is this collected sap will be getting fermented with the help of naturally occurring yeast. So this naturally occurring yeast that will ferment the sap which is collected from palm. Which palm? Caryota urens. That is a scientific name. This and all you should note down for NEET exam purpose. For board exam, what you should write? Toddy. That is a traditional drink found in South India. So what they will do? From the palm, they will collect the sap in pots and all, and for next day they will keep it. So next day, if they are cutting it and keeping, the sap will be getting collected into the pot, and they will collect it in the next day. So up to that, it is exposed, right? So the naturally occurring yeast that will help in the fermentation of that sap, and that forms the toad. Okay. So this toddy, it will be left for few hours, and that time it will undergo fermentation with the help of naturally occurring yeast. Then this can be used. Okay. Then some other microbes in some other food items like fish, to ferment fish, soya bean, then bamboo shoots and all bamboo shoots and all for what, to make some kind of food products. And for these and all, some kind of microorganisms are used. Then another one is cheese. Okay, another one is cheese. Another product is cheese. What is that? In cheese making, microbes are used. Microbes have a very good role in cheese making. What is cheese? Cheese means it is the concentrate of milk. Okay, cheese is the concentrate of milk, and it's partially degraded. Okay, it's not fully degraded. Milk is not completely degraded. Partially degraded concentrate of milk, which is partially degraded. This is partially degraded concentrate of milk. And how it is? See, in our period under, cheese means most of the people won't be knowing. So in nowadays it's available, right? Everywhere it's available. So everybody will get a taste of that cheese, right? So concentrate of milk which is partially degraded. So in cheese making, microbes have a very good or very important role. So cheese, you can in old days and all, abroad people, they were using all these items, but nowadays everywhere all are using this cheese items and all. So now it's a common food item, right? It's a very common food item, and it is the oldest food item also. Okay, in our place, it's earlier days it is not common, but now it's a common food item everywhere. So here you can see it's partially degraded, we said, and what and all present in that? Fat. In this cheese, what and all? Milk fat. Okay, milk fat and casein. What is casein? Casein is milk protein. This milk fat and casein is milk protein. Which protein is? Casein is a milk protein. Okay, casein is milk protein. So milk fat and milk protein, that is partially degraded concentrate of milk fat and milk protein, that is cheese. Okay, then you can see different types of cheese or different varieties of cheese by their texture. Different types are there, different flavors and different tastes, and based on the microbes which are used. Okay, so different varieties of cheese, they are there. For example, you can see Swiss cheese. Okay, Swiss cheese. Swiss cheese, what is that? It is formed with the help of which one? Propionibacterium shermani. Okay, write down this name. Propionibacterium shermani. Okay, so Propionibacterium shermani. So hope you know this rule while writing the scientific name. First, the generic name you should start with the capital letter, and species specific name you should start with small letter, and you have to write it and underline it separately. Hope you know that. While typing, it should be in italics. The scientific name should be in italics. So while writing, what you should do while writing, both should be underlined separately. Okay. So the cheese, there are different types of cheese based on their texture, flavor, taste, and all. So what is with the help of which microorganism? Propionibacterium shermani. This is the microorganism which is helping to make Swiss cheese. The Swiss cheese is having large holes. Large hole. Okay. So it is a large hole Swiss cheese. It is ripened with the help of which bacterium? Propionibacterium shermani. In this chapter, a lot of microorganisms name, their uses, everything will come. You have to note down each one for MCQ. That means neat purpose, especially for neat purpose. You have to by heart all these things because so many questions are there from this chapter. So these and all you can easily get. There is no application type questions. Directly you can score marks. Then Roquefort cheese, which is next type of cheese. This is Swiss cheese. This is first type. Next, second type of cheese is Roquefort cheese. Okay, this Roquefort cheese, it is ripened by which one? It is ripened by fungi Penicillium roqueforti. Okay, Penicillium roqueforti. This is the, Penicillium, it is a fungus. So Penicillium roqueforti, this is the roqueforti cheese ripening organism. It's a fungus, Penicillium roqueforti, that is a fungus which is helping to ripen the cheese. Penicillium roqueforti, Roquefort cheese, that is helping to ripen which organism? Penicillium roqueforti, that is the fungi which is helping to ripen Roquefort cheese. Next is Camembert cheese. Okay. Third one is Camembert cheese. This is the third type which is ripened by Penicillium camemberti. This is not easy to remember because Penicillium, both are ripened by Penicillium species. First one is Penicillium roqueforti because the Roquefort cheese it is ripened by Penicillium roqueforti. Here Camembert cheese, that is ripened by Penicillium camemberti. It's connected with the name, so it's easy to remember. Okay. So when this, what is that, Swiss cheese, when it is made or during the production of this Swiss cheese in large amount, the carbon dioxide by the bacterium, which is the bacterium helping for Swiss cheese, Propionibacterium shermani. So while forming that carbon dioxide, large quantities of carbon dioxide is produced during the production of this Swiss cheese. Then Roquefort cheese and Camembert cheese, both with the help of Penicillium species. Okay. So that gives Penicillium species. So that is by a fungi. So that is a fungus. Which one is helping for the ripening? The fungus is helping for Roquefort cheese and Camembert cheese. So both are with the help of fungus. Which fungus? Penicillium species. One is Penicillium roqueforti for Roquefort cheese. Then another one, Penicillium camemberti, that is for Camembert cheese. So both are fungal microorganism which belongs to group fungi. Okay. Next, we are going to learn about, from this part, NEET exam purpose. NEET exam purpose, you have to learn okay, for NEET exam purpose, you have to learn some more points that we will discuss in the last class after completing this one. Next, microbes in industrial products. In industrial products, what and all you have to learn? You're going to learn fermented beverages, then antibiotics, in the production of antibiotics, how these microbes are helping, then chemicals, enzymes, and bioactive molecules. In production of all these things, how or what role these microbes are having. That's what we are going to learn under microbes in industrial products. So in household products, we have learned about curd, then dough. In bakery items, how it is helping, how it is making, helping in the curd formation, then batter formation, everything we have learned. In cheese, all those things are coming under household products. Next, so when they are asking about in household products, where and all it is useful, which and all you need to mention, you have to mention about the batter, curd formation, then dough formation, then the baker's yeast you have to mention as an example, then cheese making. Okay, they may ask what is baker's yeast, Saccharomyces cerevisiae, that is baker's yeast. Then they can ask the curd forming bacterium, then microbes, what is microbes? So you may get such kind of questions. Okay, then they may you may get a question like the fungus which is helping in the Swiss cheese formation. What is the reason for large holes in Swiss cheese? What is the reason? The carbon dioxide release. Okay. Carbon dioxide. So what is the reason? Large and the fungus which is related to that for the ripening of that Swiss cheese and cheese. Different batches, what we have many cheese they will ask. So these are another okay, or how the batter is getting fermented. Industrial products, you have to remember to synthesize many number of products, right? N number of products are getting synthesized. So with all these products which are useful for human beings, they are getting synthesized with the help of these microbes. Okay. So here beverages and antibiotics, they are the examples. You can see some beverages and antibiotics as an example for the synthesis. Then chemicals, enzymes and bioactive molecules, all these are coming under in industrial microbes in industrial products. Okay. So in industrial scale, when you are producing microbes, industrial scale production of microbes, so that requires large vessels. Okay, that requires large vessels. So these vessels, they are called as fermenters. So industrial production of large vessels, they are called as, these vessels are called as fermenters. So what are fermenters? Fermenters, industrial production of these useful products for human beings, that requires growing of microorganism in large vessels. So during industrial production, how you will grow microorganism in large vessels, you can grow microorganism. Okay. So microorganism can be grown in large vessels during industrial production. So that is called as fermenters. Okay. Then next, what we are learning in this fermented beverages, we are going to learn about. So what are fermenters? The large scale production or the industrial production of microbes in a very large vessel, that is called as fermenters. Okay. Then fermented beverages. We are going to learn about fermented beverages. In fermented beverages, mainly we can see yeast. Yeast is used for the production of beverages. Most of the beverages like wine, beer, whiskey, brandy, rum and all. Which one you are using? Fermented beverages. Wine, beer, then what is next? Whiskey. Brandy, then brandy or rum. In all these things and all, which one is used? Especially yeast, it is used. Okay, Saccharomyces cerevisiae, which is used for bread making, and that is commonly called as brewer's yeast. Okay. Brewer's yeast. So Saccharomyces cerevisiae for the production of beverages like wine, beer, whiskey and all. For this, they are using the same yeast, Saccharomyces cerevisiae, which is used in bread making, and this is commonly called as brewer's yeast because this is used for fermenting malted cereals and fruit juices for production of what they are producing after fermenting? Ethanol. Okay, ethanol is getting produced. Okay. So yeast species, mainly which and all the yeast species which is used for alcoholic fermentation, they are Saccharomyces cerevisiae, which is commonly known as brewer's yeast. Then mainly malted cereals, molasses and fruit juices, which is produced or which is producing this ethanol. After fermentation, which product it will give? It will give ethanol. See, for ethanol production, molasses, that is the major substrate. Okay, when molasses is the major substrate for the ethanol production. Got it? So we have to discuss so many things under this. Microbes in industrial production, fermented beverages. So I'm just starting that industrial scale of production that need to grow the microbes, large scale production of these microbes in large vessels, that they are called as fermenters. So this fermented beverages, which are all the fermented beverages? Wine, beer, whiskey, brandy and all. So with the help of which one? Saccharomyces cerevisiae, which is used in bakery or baker's yeast, which is used in the bread production. The same thing is using here. That is also called as brewer's yeast. And ethanol is the end product. So molasses, that is the major or main substrate for the ethanol production. Got it? Then there are different types of this beverages. In this wine and beer, this you can see low alcohol percentage. Alcohol percentage is low. In wine and beer, you can see very low alcohol percentage or alcohol concentration is very less. So these are produced without distillation. Okay, these are without distillation. Without distillation, it is produced. Okay. Then whiskey, brandy, rum and all, they are produced by distillation, and by the distillation of what? Distillation of fermented broth. That is having high alcohol concentration. Whiskey, brandy, rum and all, it is having high alcohol concentration. It is produced with the help of distillation of fermented broth. Okay. So the naturally fermented alcohol, in that the alcohol naturally fermented beverages. In such beverages, the alcohol concentration or alcohol percentage is usually less. But in distilled, the percentage and all we'll discuss in the next class. The percentage, another point of view, the percentages in each one, like in alcohol or alcohol content, you need to learn for NEET exam purpose, that I'll discuss in the next class. After this, you want me to? Do you have any doubt?
No, ma'am.
Okay, so after this, everything, I think everything is clear. So next class, we will discuss everything, fermented beverages in detail, then antibiotics, chemicals, enzymes and all. And neat points from this part that I'll be giving you in the next class. Go through this. Thank you.